Hi again, this is Dr. Wiseman. Welcome to Chapter 3, where we talk about the phenomenon known as compartmentation. You guys will spend a fair amount of time learning anatomy, whether it's basic anatomy or specifically gross anatomy and so forth. We often talk about form and function and so forth. Well, one of the fundamental roles of anatomy is compartmentation. So when you take that anatomy course, I would challenge you to look at the structures and see how those structures serve the role of compartmentation, certainly membranes, but also solid organs, things like or the stomach or the lungs or the pancreas. There are lots of liver, lots of things. So let's get started. The first question I have for you, and I actually have two questions here. Why is this not possible based on what we know about physiology? So we have your typical not so scary zombie here. You know, zombies are a very popular motif in modern culture and movies and so forth. I was recently at Universal Studios and they have a big Frankenstein monster, set up, lots of zombies and so forth. Why is this not possible? Now, magic aside, we're not gonna go down that road. But why is this not possible based on what we know about physiology? And let me follow it up with a second question that maybe ties it into something what we would say is a little more biomedically relevant. What do zombies have in common with the items below? Gunshot wound, knife wound, soft tissue injury, hemorrhage, which is blood loss, burns, especially burns that are, say, second or don't want to, or third degree types of burns, life-threatening type burns, or even solid tumor cancers like pancreatic cancer or stomach cancer, thyroid cancer, those types of things, as opposed to, say, lymphomas or leukemias. Why would solid tumor cancers have something in common with zombies? And it's not because of mind control. Certainly, a gunshot wound doesn't control your mind and so forth. So, what do they have in common? What they have in common is a loss of compartmentation. If you do not regulate things very, very carefully in terms of compartments, you lose function, you lose the ability to sustain homeostasis, you lose life. For instance, we don't just pour gas into the cabin of our cars, right? We don't just pour it there into the driver's seat and then get going. We put it into a very, very specialized set of tubes that take it down to a tank, which connects it with a very, very specialized set of tubes to go to the right chamber at the right time in the cylinder cycle so that when the explosion occurs, it A, doesn't damage the engine, and B, creates motion that we can then use to move the car and move ourselves and the things inside our cars. That is the essence of compartmentation. We have to be able to direct energy and molecules and structures in such a way as to maintain homeostasis. That's the key of this. So I've beaten this to death a little bit, but let's move into this now. Compartmentation and spatial regulation in a larger sense is what I would call a scalable concept. That is to say, we can certainly see it at the macro level with different cavities in the body. And if you've taken 305 or a gross anatomy course or anything along those lines, you've talked about different compartmentations like the pericardial space or the pleural space and so forth. Those are what I would call macro or anatomical compartments. And that's really not where we're going to spend most of our time in this class. Most of the time we're going to be where the action is, which is at the tissue and cellular level. That's where a great deal of the body's compartmentation actually exists. If you consider the fact that 2/3 of total body fluid, total body water, 2/3 of it are actually inside your cells, that makes the intracellular space the major compartment in the body in terms of fluid volume, which is where most drugs end up. They very rarely end up in space-filled volumes unless you're inhaling it. In any case, it's at the tissue and the cellular level that we see lots and lots of compartmentation, and that's where we're going to spend most of our time. But if you were to sort of apply the compartmentation model to an extra-organismal, then certainly things like our borders and political boundaries and social boundaries are all examples of compartmentation in order to facilitate regulated interaction. The goal of border crossing boundaries is to prevent unwanted exchange, and that's the same story when it comes to either the cellular, the tissue level, or the whole body. A scalable concept. If we're going to talk about compartmentation at the tissue and cellular level, there's an important concept that we need to get down before we go too far, and that's the idea of a luminal space. The lumens of either hollow organs or in some cases hollow tissues is a very, very important aspect of what we call polarity in tissues. Polarity, if you think about polar, it has a plus and a minus. Or if you think about the North Pole versus the South Pole, the same story is true in tissues, that we have a polarized tissue structure. That is to say, one side of the tissue is very, very different than the other side in terms of function, protein concentrations, and whatever is moving across it. We call those polarized epithelia. And more often than not, all epithelia are polarized. So we need to understand what luminal means. In hollow organs, the hollow space itself is the lumen, so it doesn't have to be external space. For instance, the lumen inside the intestines is actually external environment to a certain degree, because the tube that runs basically from your mouth all the way to your rectum is external environment. It's one continuous tube. But it doesn't have to be external environment. Like, for instance, the interior of blood vessels is the lumen. The interior of the heart. The interior of the lungs is also theoretically external environment because you're breathing in air. Like I said, not always internal environment, but we're looking at some sort of a tube-like space, either where liquid or gas moves, either inside a tissue or inside an organ, if you will. And here's an example of the digestive tract, so forth. So you got to understand luminal, because we're actually going to get to a concept called apical. And oftentimes apical and luminal are synonymous. That when we have a lumen, the apical surface of an epithelium is going to be facing that hollow space, that lumen. So apical and luminal. Back to the idea of a polarized tissue structure. We have functional compartments, certainly extracellular and intracellular. The extracellular fluid actually has at least two separate compartments. We have interstitial fluid, and there are actually some subsets of that, like for instance cerebral spinal fluid and so forth, or lymphatic fluid. We also have blood plasma, which usually has a lot more proteins than most other fluids, but we'll talk about that later on. And then we have our intracellular fluid. Remember what I said, though, in Lecture 1, that the extracellular fluid tends to be high in sodium and chloride, and the intracellular fluid is high in potassium and low in sodium and chloride. So make sure you keep those in mind, because we're gonna be talking about that. Compartments, some of them are separated by membranes, like the anatomical ones in particular, and these membranes have multiple cell layers, often different types of tissue involved in that membrane. You might have epithelial layer, and then you might have connective tissue, even more than one kind. When we talk about membranes also, we might be referring to subcellular structures like the plasma membrane or an organelle membrane or the nuclear membrane. Those are all membranes, but in this case, the membrane is composed of biomolecules rather than multiple cell types and extracellular matrix. They're composed of things like phospholipids, so forth, and proteins. So phospholipid bilayer, often with lipids like the phospholipids, and then proteins and carbohydrates in some cases. So anatomical membranes, the sort of macroscopic ones, your skin would be an example of an anatomical membrane as opposed to the microscopic phospholipid type membranes. Both membranes, again, the idea of a scalable concept, involve physical separation and regulated exchange, the prevention of unwanted exchange. And we also have to be able to communicate. There has to be information flow across that. If you have a membrane which either materials or energy cannot move across, then it's very, very difficult to communicate. There's also this idea of structural support. So we have these structures called cytoskeleton structures, and we'll talk about those a little bit. But it's more than just holding rigidity or structure of a cell. We also have to have the ability of cells to stick together, but back to that idea of communication. Many of these specialized junctions not only provide support and structural roles, but also provide communication roles. And I want to begin to show you some examples of what I mean by some of these points. So let's pull up a phospholipid bilayer membrane. Again, this is a facsimile. It's usually a lot more protein. It just depends on the membrane. Different membranes have different functions, and then that means they'll have different constituencies. So physical separation, right? External environment, internal environment. Sodium and chloride, for instance, and potassium, for instance. And notice that this membrane is polar. We have carbohydrates, and those are almost exclusively found if we have carbohydrates like phosphoproteins and carbolipid type conjugated molecules. They're almost exclusively localized to the external environment. The only major carbohydrates that we see inside the cell are glucose and glycogen, or simple sugars, mostly glucose, and then the polymers that we use to store glucose, and that's glycogen. Those are the only two structures we typically see inside. So if you see carbohydrate chains on any type of a picture or a diagram, you'll know that that's the extracellular surface. Many of you probably have type A blood, and some of you probably have type B blood, and some of you, again, might have type O blood. O positive, O negative. The A and the B groups and the positives in blood are all types of glycoproteins. We call them antigens because they do elicit immune responses in the right situations, but they are basically like a name tag for a cell. The carbohydrates serve as special communication role in identifying self versus non-self in the immune system. So there's communication. We've talked about physical isolation and regulated exchange. We've got channels to allow things that normally wouldn't pass, or carrier proteins. Structural support we'll get to a little bit later. It's hard to see in this picture, but many people can envision that being the case. And here's what it's more like. It's a lot more complicated. It looks a lot messier, but it has a lot to do with those surface markers on the membranes, how we keep the regulated exchange going on. Just a little bit of review about cell membrane composition. And we have lipids, and the two major groups of lipids are phospholipids and cholesterol, which I want to talk about later, and you should be able to identify these. Phospholipids have a polar head group and a lipid tail that's nonpolar. Cholesterol is a very nonpolar molecule, and it serves a very, very important role. We often think about cholesterol as a bad thing for things like heart and vascular disease, but cholesterol, we could not live without it. In fact, we manufacture it ourselves if we don't have it in our diets. Oh, one more thing. You might see in the book about a sphingolipid. Don't worry about that. It's just another type of lipid that's important for determining specific functional areas in a cell. Certainly have proteins. We have peripheral proteins that are kind of associated with the aqueous heads of the phospholipids. And we have what are called integral, or if you happen to be from the UK, integral. That means that that either part or all of the protein is penetrating into the hydrophobic core of a membrane. Proteins that pass all the way through, we call them transmembrane. Some peripheral-like proteins are actually integral because they are using lipids to hold them down. Peripheral proteins more often are just associating with the head groups on the periphery of the membrane, or perhaps a cytoskeleton, but these integral ones actually have a part of them integrated into that hydrophobic center. Also carbohydrates, and I mentioned glycoproteins and glycolipids, almost always on the exterior side, the luminal side of the membrane. So we see them on the apical side, or we see them towards the lumen, or in other cases, just the exterior. Like in a red blood cell, it's all exterior, so it's all going to have glycoproteins and glycolipids. And here's just your stick figure. Again, polar head group, hydrophobic tail for the phospholipids. Here's a glycolipid. It has a lipid tail and then a carbohydrate associated with it. And here's another one over here. Here's a transmembrane integral protein, and here's a peripheral protein. Again, a very rudimentary figure, but here is cholesterol, and you can recognize cholesterol because of that 4-ring carbon structure. You should be able to recognize that from a mile away, that it is a steroid. It has a steroid nucleus, and so we see these things embedded. What cholesterol is doing, I might be getting a little bit ahead of myself, but I want to just say this now, is cholesterol is serving, and write this down, it is serving as a fluidity buffer. If we talk about homeostatic window of variables, one very important homeostatic window is the fluidity of our plasma membranes. If we get too cold, the plasma membrane will start to become like a crystal. If we get too hot, the plasma membrane might start to dissolve, and then we have unregulated exchange. In the case of too cold, we can't exchange anything because those nonpolar groups start getting really, really tightly packed. Okay, but if it's too hot, they spread out and it's too wiggly, and now we have unwanted exchange. And that's why body temperature is a regulated variable, and cholesterol assists with that regulation. It helps prevent the membranes from disassociating when it's too hot, and it helps prevent the membrane from crunching down when it's too cold. And here's just another nice picture. It's including some cytoskeletal elements now, which is very, very much the case when it comes to intracellular. But if I had a goal for you, I would say you should be able to recognize the outside versus the inside, not because of the words. See lots of carbohydrates on the outside. See these little green chains here and the little blue chains and the brown chain here. What do they all have in common? They have these little dot-like structures. Those are simple sugars that have been polymerized. So we have a glycoprotein, and here's another glycoprotein. It just happens to be a transmembrane. But here, those little steroid nuclei I was talking about in the last slide, there's our cholesterol, our fluidity buffer, our phospholipids, and our proteins. They're calling this a peripheral protein. That is a mistake. This is in fact a integral protein. This surface protein is in fact a peripheral protein under our definitions. It's not perfect. There are different labs and different people that might say different things, but for our purposes, this is an integral protein. This is a transmembrane integral protein. Okay? Don't see any lipid anchored ones here. Great link though, by the way. Okay, so where are the lipids? Phospholipids, cholesterol. Okay, where are the proteins? Well, we have a blue one here and a brown one here. If there's any other, there are other ones, but you can spot those. And then where are the carbohydrates? Should practice that a little bit. Okay. And then what last question I'll put is, where is the external luminal surface? Out here, in here. Now the words are giving it away, but you should have been able to do that if you're doing this right without those words. And this is just from your book. It's just showing you two different versions of basically the same thing. This protein is wiggling back and forth 7 times through the plasma membrane. And again, showing different. Now this one's saying lipid anchored. Okay, good. It's got lipids in there. It's an integral protein. It's kind of sticking into that hydrophobic layer. Here's a transmembrane. Oh, here's a peripheral protein. It's associated with the head groups, but it's not really on the head groups. It's actually on that protein. So it's close to the membrane, but it's not even bound in that case. Cholesterol and so forth. More detailed, here's our polar head group. These things are not homogeneous. In fact, different plasma membranes have different functional groups on the polar head. So polar heads can actually vary. We have the hydrophobic fatty acid tails. Again, heterogeneous. we have different kinds of fatty acids that are used in those tails. And again, it will determine the nature and the constituency of the membrane. With phospholipids, we actually have three different cellular structures that we see phospholipids present. We see them certainly in the phospholipid bilayer. We can see them in a monolayer like micelles, which actually we use for absorbing lipids in the digestive tract. And then we have this sort of cell membrane-like, it's smaller than a plasma membrane, but it has the same similar structure. It's called a liposome. It has an aqueous interior. And many of you who might be using makeup on a frequent basis might have seen different types of cosmetics that use liposomes to deliver cosmetics in or around the skin. Liposomes serve as transport vesicles inside the cells and other functions, and we are also using them in sort of novel drug delivery paradigms. But it's a phospholipid bilayer. It's just in a more of an organelle-sized vesicle as opposed to that massive cell membrane. Here's our cholesterol again. Again, we have 1, 2, 3, 4, and if you take organic chemistry, A, B, C, D, you should be able to recognize that carbon backbone from a mile away. This is called a steroid nucleus. That is to say, it's the heart of a steroid molecule, of which cholesterol, which means liver steroid, okay, manufactured in the liver, a lot of. Cholesterol is one of the important ones. It serves as a precursor for lots of hormones, the steroid hormones. Heard about anabolic steroids? Those are steroid hormones. Estradiol is a steroid. Testosterone is also a steroid. Very important. And all membranes are not the same depending on their phospholipids and their protein content and their carbohydrate. This is an idea of structure and function. So for instance, a red blood cell is meant to transport gases, but it also serves a very, very important immunologic and homeostatic role for clotting. So it has a fair amount of protein in its plasma membrane, a fair amount of lipid, but also has a lot of carbohydrate. Those are those antigens like the A groups and the B groups and the RH factor that gives us plus and minus in our plus and minus of our blood type. Myelin, on the other hand, is a protein that is found in neurons and nerve cells, nerve tissue. Doesn't serve very much of an immunological role. It's more about insulation, and so we see relatively little protein, a ton of lipid because it serves a very important electrostatic insulator role. A little bit of carbohydrate. Again, it's outside of most cell membranes. So it's an extracellular type membrane. So there's a little bit of carbohydrate there. Again, self versus non-self and appropriate neural patterning. Then let's go inside the cell. Let's go way down in there and look at our inner mitochondrial membrane where we're going to be manufacturing ATP and doing the citric acid cycle. A ton of protein. I mean, it's like bricks. If you think of bricks and mortar, you have mostly brick and a little bit of mortar. Mostly brick and a little bit of mortar. That's because we're doing lots of metabolic function. And zero carbohydrate. You don't really do self and non-self inside the cell. It's not that important. In fact, some viruses take advantage of that fact. In any case, so we don't have any carbohydrate. Why? Because that inner mitochondrial membrane is not exterior to the cell. It's not facing a luminal space in any real sense. And so it doesn't have much carbohydrate. And just bringing back a little 165 here and just bringing it in terms of the idea of compartmentation. Certainly, we have intracellular compartments in addition to the intracellular compartment, things like organelles and so forth. If you ever hear of inclusions, inclusions are generally functional structures inside the cell that don't have a membrane. Lipid droplets, glycogen, and ribosomes are all examples of inclusions that lack membranes inside the cell. But organelles are creating additional compartments, facilitating additional functional regulation inside the cell. Now, there is another type of compartmentation that doesn't necessarily involve membranes, but it does involve intracellular regulation, and that's the cytoskeleton. Now, the cytoskeleton has a lot of physiologic roles. Certainly cell shape, very, very important. Internal organization, that's the compartmentation I'm talking about. Intracellular transport, same story. And then we also can do assembly, that's sort of back into cell shape, that sort of structural role. So we have structural roles, strength, communication roles, function, and compartmentation. There are three major families of intracellular protein fibers or cytoskeletal elements, and they're generally categorized by the type of protein that they consist of. The type of protein then dictates the size, if you will. So we have at the top, so we have the name of these things: microfilaments, intermediate filaments, and microtubules. We have the type of protein that they're made from. For example, microfilaments are made of actin. Intermediate filaments are made of keratin, which we find in our skin and our hair, in our fingernails. And then thirdly, we have tubulin, which makes up a microtubule. Some basic functions. We see cytoskeleton helps with cell shape. It associates with myosin, very, very important for muscle contraction. Intermediates also can do cell shape and that sort of thing, but where we see it is in hair and nails, defensive purposes, for the external part of the body, protective barrier of the skin. It's a watertight, it can form a watertight mesh, which is what your skin is. Microtubules are very much intracellular movement or whole cellular movement. If we have cilia and flagella, that's involved in cells moving around. We only have one type of flagellated cell in the body, and that's a male sperm. That's the only human cell that has a flagella. There are cells that have cilia, though. That increases their surface area for absorption and transport. We also use microtubules for mitosis, but also intracellular transport. I'll show you a video probably during one of our live class sessions on that intracellular transport, which is very important for spatial regulation inside a cell. And here are some examples of cilia. You know where you see ciliated epithelium is in the fallopian tubes. So very, very important for lateral movement of either an unfertilized or fertilized egg towards the uterus. Super important. Cilia are very, very important for increased surface area inside the digestive tract and absorptive surfaces. Cilia are also important for airways. They move particulate matter out of the airways up towards the epiglottis. You ever have to clear your throat? What you're doing is material has arrived by ciliary motion to your epiglottis. You clear your throat with a little bit of muscular action, and then you swallow it. Okay? Good for your immune system, by the way. But we're using cytoskeletal proteins. Flagella, cilia, both use microtubules in specific ways. Are in context a little bit. There's not a whole lot here I can show you, but just showing you the examples of where we find things. We find microfilaments often on the interior surface of the plasma membrane. We call that a cortical actin, and it forms a meshwork just inside the plasma membrane. We certainly see it in skeletal muscle, and we will see it in other areas. I'll make sure I point it out when we see pictures. As part of the ability to regulate things spatially, we not only have to have cytoskeletal proteins, we also have to have proteins that interact with those cytoskeletal elements. And we have three major families. Like three families of cytoskeletal elements, we have three families of what we call motor proteins. We have what are called myosins. Myosins tend to interact with actin and microfilaments. Okay, so when we have actin and myosin in the skeletal muscle, or if we have smooth muscle, that's also actin and myosin now that I think about it, or if we're doing some moving things around the cortical surface of the plasma membrane, we will use myosins to do that. We have another family closely related. They're called kinesins and dyneins. These guys don't associate with microfilaments. They associate with microtubules. Oftentimes they have a tail that is hooked up to either a vesicle or another molecule, like what we call cargo here. And I have another really cool YouTube video for you that I'll show you during one of our live sessions so you can see these motor proteins at work. Motor proteins are like the Star Wars walkers. They literally walk along a microtubule in a given direction. Microtubules, like plasma membranes, are polarized. There's a plus end and then there's a minus end, or a positive and a negative end. Dyneins move towards the minus end. Kinesins move towards the plus end. So they will only move in a given direction on a microtubule. Kinesins and dyneins. And I have them there. I should probably just go ahead and dyneins are involved not only in movement of vesicles, they're also involved in the flagella and the cilia. So that ciliary motion and that flagella motion I talked about are all dyneins, but they're part of the same family here. Here's just another schematic of a motor protein. This would either be a kinesin or a dynein because it has that cargo here. And depending on whether this is the plus or the minus, if it was the minus, we would be a dynein. It hydrolyzes ATP, and that causes a walking-like motion that we're going to actually look at in greater detail, especially when it comes to myosin. Now let's start putting these pieces together and talking about tissues and how tissues then become organs. What is a tissue? It's a mass of similar cells that perform a specific function. Often, maybe even more than one cell type in some cases. Many tissues, though, are somewhat homogeneous, but there are heterogeneous. And we also have organs, combinations of tissues and cells, again, a specific function, usually a higher degree of complexity. So let's go into this. I know I'm running a little bit long, but I'm going to get us through the next 4 or 5 slides, and then I'll call it a day, and then we'll pick up with part 2 with the different types of tissue. And so we've talked about this idea of assembling and collaborating. And for assembly and collaboration, we certainly have to have communication and the appropriate structure and organization. If we lose either of these, we are going to lose function. Look no more than somebody who has neural damage. They lose that cell-to-cell communication and now they have muscle weakness. Or somebody who has a burn and they can no longer retain fluid in their internal compartments. They start to lose fluid out. Dehydration is one of the major causes of burn deaths. When people have significant burns on their body, they lose the structure and organization of their skin and they begin to lose fluid to the external environment. But you can see examples of this, and there's a really nice one here. Dr. Kevin Gribbins in our department teaches a really cool course called Histology. I'm kind of jealous. I'd like to teach that course. But he talks about these different tissues and organizations, and you can see we've got a certain organization here. This is a secretory gland, as you can even see the lumen here a little bit, but very organized tissue. Slightly less organized tissue surrounding that lumen, or that organ or gland, might be connective tissue. And we can certainly see that here. We have very organized tissue over here on the left, a little basement membrane. That's an epithelium. We have maybe some connective tissue. The cells are further apart out here. Maybe another gland of some kind, like a sebaceous gland. This is skin. So we have the epidermis, multiple cell layers. We also have that connective tissue and then some other tissues. Again, communication, organization. And I'm going to stop right there for now. We will pick up on the 4 different major tissue types in human systems in part B of chapter 3. Talk to you soon.